joining us today. First off, we just want to thank Sail Therapeutics for taking the time to meet with our community and share their expertise on what is currently the only treatment for PDCD, sodium dichloroacetate, also known as DCA. And by only treatment, I just want to clarify what I mean by that. We all know that the ketogenic diet can be used as a treatment, but unfortunately, until we have a standards of care documentation, newborn screening committees and others do not recognize it. So I just want to clarify that. I'd also like to thank SAIL for their openness to share an overview of the clinical trial and the current process with the Food and Drug Administration. Finally, and most importantly, we'd like to thank them for their continued dedication to this treatment for our community. And I just want to point out that not all biotechs would continue with the program upon receiving a CRL from the FDA. So we're very grateful for that. Secondly, I want to thank all of you, the PDCD community, the parents, the caregivers, the patients, and the physicians for your attendance and your participation today, as well as those of you who have immediately jumped into action and have already shared your testimonials, circulated the Ignite advocacy forum for Congress, contacted your elected representatives and local media. The momentum and the unity we have in our PDCD community cannot be overstated, and I know SAIL is grateful for that too. I'll go over a little bit of housekeeping. So just a reminder that this is a 1-hour webinar. We ask that you do not personally record the webinar on your end today, as SAIL is in continuing dialogue with the FDA. Hope for PDCD will post a recording as a follow-up to this webinar once we have that available. We'll also have time after the overview of the trial for questions, and then we'll close with calls to actions and next steps that we can all take. And finally, the last message I want to leave you with before I introduce everybody and we move on to the DCA testimonials is that receiving a CRL is not the outcome we were hoping for, but we, the PDCD community, are not powerless in this situation. We have the power to make our voices heard. Today, you'll be hearing from Dave Pinacki, the CEO of SAIL Therapeutics, Kyle Ashton, Head of Medical Affairs, Sharon Ham, Chief Development Officer, and as well as Brian Nappy, Chief Strategy Officer. I'm going to hand it over to Kim Higbee, one of our board members on Hope for PDCD, to share testimonials from parents who have actually received the DCA treatment for their child and the impact that that's made on their lives. Well, we already went over the meeting agenda.
Thanks, Francis. Okay, we were gracious enough to receive 3 testimonials from some of our PDCD community members. The first testimonial comes from Lisa, and it's regarding her daughter, Lexi, and this is Lexi's on DCA. So what DCA means to the PDCD community. Lisa says, my 18-year-old daughter, Lexi, is one of the oldest participants in the trial. She has been part of it since 2022. Her quality of life has steadily improved since she entered the open label phase and began taking DCA on a steady long-term basis. Prior to starting DCA, Lexi suffered from poor appetite, failure to thrive, frequent vomiting, severe ADHD, and sleep disturbances. Lexi was unable to focus on activities for even a short period of time. She often had to be removed from her classroom because she was unable to sustain attention to a task. She had great difficulty falling asleep and regularly woke up during the night. Since Lexi started taking DCA, she has been able to stop taking her ADHD meds and sleep medications. She is now able to sit still and participate in school and social activities. She no longer runs out of her classroom due to an inability to focus. She falls asleep on her own and sleeps through the night. Lexi no longer has vomiting episodes, and she has grown several inches and gained weight. For the first time in her life, her height and weight are on the growth chart. She is able to eat a wide variety of foods. Thanks to DCA, Lexi now holds a job, has friends, and lives a relatively typical life similar to her peers. This is an amazing DCA story.
Can I just interrupt for a second? Some people are having trouble hearing sound and some people aren't. I wonder if the old registration link is different than the new registration link, or if there's something we can do.
The links, but it was nothing changed on the actual link.
Okay. Someone said they can see the text, but they can't hear, and other people said they couldn't hear. But you can keep going. Maybe just if people can't hear, let me know in the chat and we can figure it out.
Okay. All right. I'm going to move forward with Lydia here. So Erica and Dorian submitted their daughter Lydia on what DCA means to them. Ever since Lydia started on DCA, her condition has been much more stable. Before DCA, even a small cold would cause Lydia to have flaccid episodes, losing all muscle tone, unable to even hold her head up. This hasn't happened since starting DCA. It makes her so much more stable and resilient. Since starting DCA, Lydia's mental and physical development has also accelerated. She started walking, feeding herself more independently, and gaining verbal and communication skills. We truly believe DCA is helping Lydia achieve her full potential. And our last slide was what FDA denial means to the PDCD community. And this is Abby and her parents, Nikki and Will, submitted her. To them, the denial means devastation. That's the only word that comes to mind. I've spoken with so many parents whose children are on DCA, and even the smallest improvements they've seen, more energy, more comfort, more good days mean the world. For families like ours, those small gains aren't small at all. They are everything. Losing access to DCA would feel like losing hope. When your child has PDCD, you cling to anything that gives them a better quality of life, no matter how modest. Taking that away strips away not just a treatment option, but also the chance at giving our children more dignity and joy in the short time they have. That was powerful.
So now I'm going to hand it over to Dave Panacki, who's the CEO of Sail Therapeutics. We just wanted to take a little bit of time before we get into the clinical details on what DCA means on the human side and put a face to what it means to our children.
Yeah. Thank you for sharing those stories, and thank you for giving us the opportunity to speak with you today. We're updating you on what we term as SL1009. Many of you discuss it as DCA, but that's the active drug ingredient in the medicine that we're studying. It's important to note that this is investigational. This medicine has not been approved, and we have ongoing discussions with the FDA. And so, from that perspective, we want to make sure that that's not misinterpreted. We're here to tell you a little bit about the situation, the history of the development program, and then really what our next steps are, and offer the opportunity for questions. Particularly, you may have some after seeing some of the data that we present. So I'll be starting that out. I'm Dave. I'm the CEO at Sail Therapeutics. You'll hear from other members of our team, Kyle and Sharon in particular, and then we'll all be available for questions during the Q&A session that follows. So first of all, we'd just like to thank everyone for those of us on the drug development side that are familiar with rare disease research. This is something that is really important to those that work on it. And then it requires a huge investment of time, emotion, whatever it is that's necessary in order to involve yourself or one of your children in one of these studies. And so we want to thank everyone who's contributed to that. We'd like to thank the physicians and investigators and coordinators that have also helped. And then lastly, in order to do that, you may just be a part of getting the community together and also providing more information that allows places like the FDA, like companies like ours, like the community and Hope for PDCD to better communicate the impact of the disease. So we appreciate all the advocacy efforts that everyone has done. Again, this presentation isn't intended to be shared outside of this group. Hope for PDCD and Sail are recording it. That will be available momentarily once it's done. And we continue to be in dialogue with the FDA. So as we've mentioned publicly, we hope to have a Type A meeting upcoming. We'll talk to you a little bit about what that is. And then we also hope that leads to a much faster pathway for potential approval of this medicine to make it more accessible to patients that are out there. Okay, next slide. So many of you probably have not heard of us. Our company's called Sail Therapeutics. We've been around since 2015, and we have offices. Our primary headquarters are a group of folks who work in Dublin, Ireland, as well as Atlanta, Georgia. We have around 23 employees. We have a number of development programs. This is our latest stage one and most prominent. And we've been able to fund 5 clinical trials, 3 of them using SL1009 or dichloroacetate. So, from our perspective, this is really the centerpiece of everything that we've been trying to achieve for quite a long time. And, we're hopeful that we'll be able to find a new path forward. But, we have a small number of folks that are working very hard to get this done. Slide. We have been working on this program for a long time. And then before us, the investigators that are involved, starting with Dr. Peter Stackpole out of the University of Florida, have been working on it even longer. To give you context about how long it takes to develop medicines, the initial conversation that the investigators had with the FDA was in April 2013. Okay, so 12 and a half years ago was when they started initiating the discussions to figure out how to make sure that this study was run well. We were able to enroll the first patient in 2020. So that was a really important date for the company and for the program. And then things progressed much more rapidly towards our filing for approval after that. Probably the most important other date in here is that there was a listening session. So the PDCD community actually had the opportunity to speak with the FDA. In that session, we had brave parents that came out and talked to the agency about the challenges associated with this. We had the community foundations and some of the treating physicians to support that. Information like that is foundational for companies like ours and any company that's studying a disease like this to sort of, show the FDA the importance of having a treatment option and what the implications of the disease are. So for those of you that were involved in supporting that, taking the surveys, et cetera, that started that, we really want to thank everyone that was. And then lastly, as you know, recently we were given a complete response letter, and we're in the process of trying to work with the FDA to resolve that. That was a surprise to us, and we hope that we're able to find a fast path forward at the agency in the coming weeks. Right now, we're going to share the overview of our clinical data. Kyle Ashton from Sail Therapeutics will be doing that. And, we hope that that's informative to you to understand, a little bit about the study, a little bit about the results we had, and then we can talk more about the interactions that we've had with the agency and what we hope to do going forward. Kyle?
So you go to the next slide. Thanks, Dave. So this is a broad overview of PDCD, and you all are familiar with PDCD. So I'm just going to make a few points here that have to do with doing a clinical development program for PDCD. So as you know, this is a rare disease. So if it's a rare disease, it's much more difficult to do a clinical trial than if it's a common disease because you have fewer potential individuals to participate in a clinical trial. You're not going to have 10,000 patients participating in a clinical trial like you would in like a cardiovascular outcome study, for instance, right? So that means that, you're going to have less power to detect a statistically significant difference, which makes it much more difficult to have the level of evidence that you would see in a more common disease. Another thing with PDCD, it's a heterogeneous disease, as you all are familiar with. Every individual living with this disease varies, right? So varies in terms of the symptoms they have, how frequent the symptoms are. You saw in the few cases that were presented at the beginning of just the different aspects of the daily life that are affected. What links everyone is this deficient energy production, which I'll talk about in just a minute. But I want to point out when you have a heterogeneous patient population, that also makes it difficult for a clinical development program, because if you have a homogeneous where everyone is exactly the same with respect to one particular characteristic, for instance, then it's much easier to do a clinical program and to get a statistically significant result. The other thing here is there are no FDA-approved treatments for PDCD. When there are FDA-approved treatments for a particular disease, What that means is there's some endpoint or endpoints that have been validated and that have been accepted by the FDA as valid for approval for that particular disease. And so if you don't have those, then you have to design your endpoints. And ideally, you could use a surrogate endpoint like a biomarker, like in our case would be lactate, or you develop some sort of clinical outcome measure. But as you saw in the slide before, probably with the involvement of some of the folks on this meeting, it took a few years to develop the primary outcome measure for this clinical trial, and it's a novel endpoint. So you're going into the unknown with that endpoint. So really just wanted to root everybody that it's very difficult to get statistically significant results in a clinical development program for a rare genetically based disease. That's really the point, and just a context to keep in mind. So we can move to the next slide. I will say that as we're moving to the next slide, we are encouraged that the FDA recently announced rare disease development evidence principles, which suggest regulatory flexibility for rare diseases, including not requiring placebo-controlled, randomized, double-blind controlled trials, et cetera. So hopefully that will continue to evolve and take places so that new treatments can be developed for rare diseases. This is a slide just to review because we're going to talk about survival, and just wanted to give you a couple of pieces of data from the published literature suggesting what the survival curves look like for individuals living with PDCD, just to give you a context, because later on we're going to talk about survival as part of the data that we have for the PDCD program. So we can move to the next slide. This slide has a lot on it, so I'm just going to speak through it. The takeaway here is that the mechanism of disease, how PDCD happens, and the consequences of that are well described and well known. The mechanism of action of DCA, or dichloroacetate SO1009, is also well known and well described. So how this basically works is, that if someone has PDCD, they have a pathogenic variant. It's in a gene that's encoding one of the subunits of the pyruvate dehydrogenase complex. So what does that mean? That means that you're going to get less activity of this complex, the PDC. And so if you look at the lower left there, in an individual that doesn't have PDCD, you consume a meal, you break down the carbohydrates to glucose, that gets converted to pyruvate. The PDC then helps convert pyruvate to acetyl coenzyme A, enters the TCA cycle, generates ATP. Ultimately, you produce energy, right? So if you have PDCD, that's in the middle part, you have some deficiency in the enzyme function. And that deficiency in the enzyme function means you're getting less of that throughput. So you're going to get a buildup of what's upstream, so that's pyruvate, and you're going to get a deficiency of what's downstream, which is acetyl coenzyme A. So what that means is that you're going to get less of the precursors going into energy production, so you're going to get less energy production, and that energy that you need for your body and your brain, you're going to have less of it. The other thing is with the upstream with pyruvate, you get this buildup of pyruvate that gets metabolized to lactate. And so that's why you typically see in PDCD, you see less energy production and you see a higher level of lactate. So with DCA, what it does is it inhibits this kinase, which is shown in the red X on the side of the lower right panel, upper left X. It inhibits this inhibitor of the cycle. So basically, if you inhibit an inhibitor, it's like ramping up the cycle. So what you're doing is you're maximizing how much of the PDC that an individual can do. And so by giving DCA, you get more of the glucose getting converted to pyruvate, getting passed through to acetyl coenzyme A, getting to the Krebs cycle, TCA cycle, generating energy. So what you should see in an individual given DCA that has PDCD is you should see an increased energy production, so getting more energy to the body and the brain. And then you also should see a metabolic stabilization or of the lactate pyruvate conversion. So what that means is that lactate should go down. And then in addition to the increased energy production. So that's basically the key point here is that we know what the mechanism of disease is, and we know what the mechanism of action is. So that's very good as a foundation for a clinical development program. So if we go to the next slide. Now, a little bit about dosing for SL1009 or DCA. So if you look back in the literature from earlier explorations of potential uses of dichloroacetate, what was seen is there was a high incidence of peripheral neuropathy. The observation was that it tended to happen more so in older individuals, and that gave an indication that it potentially had to do with how quickly individuals were metabolizing the drug, and potentially the older individuals that were metabolizing it more slowly were accumulating the drug, and that was leading to this peripheral neuropathy. So investigators looked at how DCA is metabolized and figured out that the gene that's responsible for the enzyme that metabolizes it is GSTZ1. And then they did haplotype analysis of individuals and can divide individuals into fast and slow metabolizers. So ultimately what this means is that before you get your dose of SL1009, say in a clinical program or expanded access program, you do a cheek swab with your clinician. They send it in and do a haplotype analysis, and you'll get a result back that says whether you're a slow or fast metabolizer. If you metabolize DCA quickly, so you're a fast metabolizer, you get the higher dose. If you metabolize DCA more slowly, you're going to get the lower dose. So this is something that was figured out prior to starting the randomized double-blind placebo-controlled trial, and will be important as you consider the results of that. So this was the first study that used this as part of the development program of the drug dosing. So we can move to the next slide. So there's a bit of a busy slide too, but just I'm going to present two phase 3 protocols to you. One is a randomized double-blind placebo-controlled trial. The other is a survival analysis. So first with the randomized double-blind placebo-controlled trial, and I'm sure some of the families on this call were part of this. It began with a screening period. Then individuals were randomized to receive either DCA or placebo for 4 months. There's a 1-month washout, and then crossed over. So if you got DCA for the first 4 months, you got placebo for the next 4 months. So everybody got a period of 4 months where they were either on DCA or placebo. And the dose that they got was based on that haplotype, which I just shared with you. At the conclusion of the randomized double-blind placebo-controlled period, there was then the opportunity to enroll in the open label extension. And we have individuals that have been in that and continue into that up to about 4 years now. We randomized 34 patients total. 28 of those continue in the open label extension. I already talked about the dosing. As far as the primary endpoint, so this was the observer reported outcome motor score. So some of you are very familiar with this. Basically, it's a daily observer reported, caregiver reported survey of their motor symptoms. So rating each individual on their weakness in coordination, their muscle tone, their rigidity, and whether they're having involuntary movements. And each domain is rated from 0, either mild or absent, to 4, extremely severe. So if you have none of this, then you would have a score of 0. If you're rated extremely severe across the 5 things, then you would have a score of 20. So the scores can range from 0 to 20 entering the study. I'll mention that we did, as a secondary endpoint, track lactate levels, because as I showed you with the mechanism of action of how this drug works and the mechanism of disease of how PDCD happens, that you should see a stabilization of lactate after treatment with DCA. So we'll go to the next slide. Thank you. So overall, with respect to the efficacy results, I'll show you that first, and then I'll show you the safety. So this was for the 4 months on DCA or placebo. The primary efficacy endpoint, so that change in observed motor score from baseline to end of treatment, there was no statistically significant difference whether individuals were treated during the DCA period or placebo period. So we then queried the data to see if there was a signal or some indicator of a benefit for at least some patients, and found a couple of things. So one is that there seemed to be a floor effect. So that basically means that individuals are entering the study with a very low score. So there's not really much room for improvement. The other thing is if you look at individuals with higher baseline scores, so let's say out of greater than or equal to 8 out of 20, so that would indicate moderately severe on the motor subscale at baseline. If you look at those individuals, it did suggest that they potentially had more of a benefit from DCA. The other thing we looked at is to see whether if you follow individuals for a longer time period, whether you potentially see improvements. And that does seem to be the case if we include the open label extension data. So the 4 months on randomized double-blind placebo-controlled trial with DCA, continuing out through the open label extension. In the intent to treat population, they went from a baseline around 7 to a final observation of 5, so a decrease of about 25%, meaning an improvement of about 25%. It was driven largely by the individuals that had more severe scores at baseline, but even the individuals with lower scores at baseline also did see an improvement. So there does seem to be an indication that with Obzero, the longer you follow individuals or the individuals that come in with more severe at baseline, that's where you potentially see benefit with this. Also mentioned for lactate, so we did see a statistically significant reduction in lactate, which is what we'd expect based on how this drug works. That you're going to get a metabolic stabilization of lactate in that conversion of pyruvate to lactate. So that's the efficacy from the double-blind period. So now if you go to the next slide. One of the key findings from the double-blind period was the safety piece, because remember, as I talked about, this is the first study that used this personalized genetic test to help determine what dose an individual should get. And in the past, there had been an issue with peripheral neuropathy. So by using this personalized genetic test to help with the dosing, so that we can dose based on how quickly an individual metabolizes the drug, whether fast or slow. What we saw was no incidences of peripheral neuropathy during the double-blind period. Now, I will say we did have one case of mild peripheral neuropathy after 18 months of open-label treatment, but essentially, particularly for that 4 months during the randomized double-blind, we saw no peripheral neuropathy. So it does seem like this genetic testing to help drive the dosing is helping with respect to adverse events. And if you look at the safety profile here, overall, it's very placebo-like. So remember, there's 34 patients that were involved in the clinical trial. And if you look at the numbers here on placebo, 1, 2, or 3 patients indicated having adverse events in one of these areas. And in the SL1009 or DCA group, it's 1 or 2. And if you look particularly in SL1009, the most frequent adverse event was 2 individuals out of 34 reported having GI disorders. 1 had constipation, 1 had diarrhea. And so what you see here from the safety profile is much improved, largely because of this, or likely because of this personalized genetic dosing with this haplotype test. So now we can move to the next slide. So the second protocol I'll talk to you about is our phase 3 study of survival. So here what we did is we compared the survival of individuals in the SL1009 program. So that included the double blind I just told you about, plus the open label extension. And we needed something to compare this to. So because we look at the published literature to see what data is available with respect to survival and tracking survival over time. And there was a study published in 2012. This is a natural history cohort, and this is what we compared it to. And in this population, there was a 39% mortality rate. And so this was the analysis that we did where we compared survival to a natural history cohort to give us an idea of whether the survival is different in our study versus what was observed in this natural history cohort. So the primary endpoint here was comparing survival. And we particularly, when you're comparing across 2 different studies, you need to match the patients as much as you can, so the data matches as much as possible. And so here we matched based on gender and age, and then we did additional analyses to see whether being on ketogenic diet or not mattered, whether changes in standard of care mattered, and additional survival analyses. So we did a lot of sensitivity analyses to see, if that affected the data. So if we go to the next slide. Okay, so on the left here are the survival curves, and then on the right is a summary of the data. So for the SL1009 treated patients, so we're tracking out to almost 4 years on drug, and the blue line is for those individuals. So what you see is that you have 100% survival through when the data was tracked. And then you compare that to the red line is the natural history cohort. So these are individuals not treated with DCA from that published study. So the primary endpoint was met. There was a statistically significant difference in survival rates between the two groups. We did 10 matching sensitivity analyses to address potential matching concerns, and all of them were consistent with the overall findings. So this seems robust to the ways that we've been able to query the data thus far. So if we go to the next slide. So a summary of the key findings. So with respect to safety, we've shown that SL1009 is well tolerated upon chronic administration of over 3 years. The overall incidence of adverse reactions was similar to placebo, and in particular, when we think about peripheral neuropathy, we didn't see any of it in the randomized double-blind placebo-controlled trial, and we only saw one mild case 18 months into the open-label extension. From an efficacy perspective, so primary efficacy endpoint, this observer-reported outcome, the motor subscale, in the randomized double-blind placebo-controlled trial did not demonstrate a statistically significant difference between placebo and DCA over the 4 months of each. However, there was a trend in individuals with higher baseline scores towards improvement. And then also, if you track individuals longer, including the open label extension, we also saw a statistically significant improvement. With respect to the key biomarker lactate, we saw significantly decreased levels of lactate during the randomized double-blind placebo-controlled trial. And then we saw a statistically significant difference in survival when compared versus an untreated natural history cohort. So that's a summary of the clinical data. And now I think with the next slide, now that you've seen that, we can transition to some of the details of the CRL vis-a-vis our data package. And Sharon, are you here for this one?
Yep. Hopefully.
Mhm.
Thank you very much, Kyle. Moving to this section, basically, we'd like to maybe help walk people through just what it means when you see CRL and how that's to be interpreted. So a CRL is FDA parlance for a complete response letter, and it's basically the FDA's way of saying we're finished with the review of the application, but it's not ready for approval. And this means basically that it can't be approved in the current form it's in. It doesn't mean that there are not opportunities for you to come back and have further engagement with them or provide additional information. In a CRL letter, it basically identifies the specific issues that the FDA found during the course of their review. These are pretty varied across the course of which application the FDA looks at, but it could be inclusive of things like a need for additional evidence of safety, additional evidence of efficacy. It could be that they had an issue with how the clinical trial was conducted. There may have been questions that related to manufacturing or quality issues for manufacturing, issues for labeling, or the like. But in short, it tells the company directionally what it is that they're looking for that needs to be worked on before an opportunity for resubmission. And sometimes, as you might imagine, these fixes are relatively small. Sometimes it's the conduct of additional study or studies, and that can be years in the running still in the future. A CRL, however, does not really mean the same thing as a rejection. It simply is a way of saying you're not ready yet. Next slide. So with that, what I thought I would do is basically walk through the issues that are specific to the sales CRL. We received our letters we've shared on the 27th of August. There were no issues identified that related to anything on the fronts of safety, chemistry, manufacturing, or quality related issues, or anything on confirmatory evidence. The real issue that was the kind of focal point for the highlight on the CRL was the clinical studies section. And you've heard some of this background now from Kyle. So hopefully this will make a little more sense. The two studies in particular that were the focal point of the application, in this case identified as study 1 for the placebo-controlled trial and study 2 for the natural history cohort. In the case of the placebo-controlled trial, they identified that if the primary endpoint had not been met, which we knew, we had identified that as well. And they also said that the way in which we assess the higher baseline patients, they just weren't comfortable with in terms of how that was assessing the treatment effect. In the secondary endpoint case of lactate, they acknowledged that the endpoint was met and was statistically significant, but they just weren't sure they knew how best to identify that correlation to a clinical benefit. In the second study, this was a survival study where, again, as Kyle highlighted, we compared the outcomes of the first study with a natural history cohort of patients. In this case, FDA was concerned that some of the matching to the external control wasn't exactly equivalent. As you might imagine, that's not an easy thing to do because you're looking at two different sets of populations and you're trying to identify them as closely as you can for a match. And the second piece was that they also identified that there may have been differences in diagnostic methods that were used at a certain point in time as contrasted with the time frame in which we conducted our study. So the combined elements or reasons that the agency has identified as areas of concern as they've reviewed the two trials that were considered pivotal to our application. Next. With that, I think, Dave, we'll shift to you and what's next.
Yeah, thanks, Sharon. So as we mentioned earlier, we're going to request a Type A meeting. That's a standard process after you receive a complete response letter. And one of the things that's great about it, and the agency, the FDA, also understands that this is an important time for companies and therapies and patients. And so this is one of the meetings in which they respond to the fastest. So it's a highly prioritized meeting. It's a formal discussion that should come within a month or so of when we request it. And it's intended to address significant roadblocks. If there are misunderstandings between the FDA and the company about some of the data and the results, address those, and then identify what's necessary to get to the next step of the development program to move towards approval. Our goal is to submit additional evidence that supports this. So there are a few places that we're doing this, just to give you an example. When we submitted the application, it was in November of 2024. Obviously, now it's September of 2025. Patients have remained on therapy, and we've collected additional data. So we have additional data from that. One of the matching items that the FDA identified, for example, had to do with information that's available from brain MRIs. We actually didn't collect everyone's brain MRIs, and we didn't have that information. So, we're working with the sites to try and get that information so that we can better answer the question. We want to do that before we go back to the Type A request. So anyone who's here and is receiving questions that may be a part of the study and receiving questions about that, if you've had those types of documents, that would be very helpful, as we'll be incorporating that into our responses to the agency. We're hoping to have the meeting in early November. We plan to continue to support patients on the open label extension. We also have an expanded access program that we had begun the process of initiating and have a number of sites up and running, and I believe a few more that are shortly going to be. And as we've said before, this meeting is really important, and success of this meeting is really important, because if we are unable to find a pathway forward, it doesn't require a future additional study, we won't be able to continue to support these programs over the long run. So for us, this is obviously a very important meeting. We want to make sure that we put our best foot forward and bring any possible additional data that we can. With that, I think that we will go ahead and have a question and answer session, to the community. I know that the team from Hope for PDCD had planned to manage this, and so I'll pass it over to them to take it from here.
Thanks, Dave. Okay, so Katie Cairo here, and what we're going to do as we move into the Q&A now is we've grouped the questions into 3 different categories. So what I'm going to do is I'm going to read through the questions for those who may be listening in and can't see on screen. So SAIL team, feel free to answer accordingly the best that you guys can, understanding some of these questions may be better suited for a physician, but we are going to try to get to as many of them as we can here. And we will be starting with the ones that were pre-submitted. So thank you guys for putting those questions in. So Francis, if you want to go to the next slide, we're going to first up start with the questions on the denial. And I know some of these were just answered as Sharon went over the CRL with everyone. But we do want to reiterate that the drug was deemed safe. So these 2 questions here, why was the approval denied? And is their denial mostly a documentation issue? Haven't kids been using this without problems? Dave and team, is there anything else that you'd like to add specific to the denial specifics?
Sharon, do you want to go ahead and take this?
Yeah, I mean, I think as we've shared, we don't think of it necessarily as a denial. Clearly, it wasn't the expected outcome that we had hoped for, but it does identify for us areas that the agency wants to better understand, and will give us a focal point for how we'll address our discussions with them in the upcoming Type A meeting. So I think that basically is how we think about it, and it's clearly not just a documentation issue as we framed it. They have very specific areas that they're just trying to better understand, and I think it's on us to try to help now pull that forward for them.
As it relates to additional documentation, we do think that there are some places where documentation will really help. Like I had mentioned, the MRIs. And so from the standpoint of, what we want to do is anything that is documentation based, we want to collect that documentation and we want to support our answers. And anything that then is more, I would say, subjective in nature about, the perception of treatment effect and efficacy, I think we want to make sure that we can focus our discussions on that with fewer of these issues that might be related to documentation.
Okay.
Okay. So Francis, I think, yeah, we can move on to the questions specific to DCA. So I will read through a couple of these here. So does the drug have any major side effects? Does this drug help decrease of seizures? What diet should be followed for this drug regimen? And also, is there enough funding for continuation of this research trial? So I know that was like 5 questions in one. So I'll let you and the team decide who wants to take maybe the first set, and then we can talk about the continuation of the funding.
Sure.
Yeah, I can kind of umbrella it. So, with respect to, who's most likely to benefit in our expanded access program, it includes anybody who has a genetic diagnosis of PDCD. So that's basically what we're looking at with respect to the potential for this program. From a diet perspective, as was mentioned early on, ketogenic diet is something that's often used in as part of the treatment plan. How this product works vis-a-vis the diet is something that I think still, could be explored further. But one of the things that's been pointed out is the potential to adjust the diet so that you can maximize the potential benefit you're getting with SL1009. So that's something that, obviously, if it comes down to individual questions about your patient or yourself, talk to your clinician about that for sure. Presented the information with respect to the adverse events that, placebo-like adverse events The peripheral neuropathy, as I mentioned, we didn't see any in the randomized double-blind placebo-controlled trial. We saw the one mild case 18 months out into open label. With respect to stopping, we don't expect and haven't seen anything with respect to adverse events that would linger. Similar, like some medications, when you take them, you have to be weaned off. That's not really the case here. And I think that kind of hits the key things here. But Sharon or Dave, anything else that you would add?
Yeah, I think maybe just a couple of extra points. Again, some of you who are involved in the trial might know this, but the majority of patients in the clinical trial were actually on a ketogenic diet. So I think it's important to know that it is in fact a baseline that was used as part of the treatment regimen. And with respect to seizures, we did do an assessment of seizures. I think the challenge for us is just that we have a small population. We saw some significant improvement in seizures in several patients, but because we have a small population, that population size was actually too small for us to be able to demonstrate that in a statistically significant way relative to the size of the total population. So, so we did see some benefit, but it was in specific patients, and it's not something that we could sum as a total population because not everyone has seizures as part of their baseline.
To access, so the open label extension continues. So individuals that participate in the clinical trial still continue to have access via the open label extension.
Okay, thanks, guys. And just for that second part of the question, I think you already answered it. If we're not able to get that type 1 meeting and move forward with the path with the FDA, that the funding for continuation would not be there for an additional trial. But is there anything else that you guys would like to add to that?
Here, I'll go ahead and take this. I mean, what I would say is, we're hopeful. The agency recently published a program called the REDP program. I think it's the Rare Disease Endpoint Program or something like that. Anyway, in that program, they discuss things like how you would treat diseases such as PDCD, typically, diseases that are 1,000 patients or less, have a known genetic cause, don't have treatments. And in that, they talked about a number of ways that they would evaluate those applications. That was posted by the FDA on September 3rd. So after we received our complete response letter in August. And so although right now the idea of initiating a new program going forward is not something that is feasible to do in the way the FDA described it to us, the idea that the agency has the mindset that they would like to work with companies like us to get something approved is there. So we're really just trying to do the best we can to come out with the best possible outcome. We know that this is an important thing for patients. We know that there are no treatment options out there, and we think we've shown a good bit of evidence that supports a potential pathway here. So that's really what we can share. What I would say is, to the extent that you can help us, those would be if there are questions related to those of you that are in the study that study coordinators are reaching out to you for, please respond to those quickly, because that draws the timeline at which we can go ahead and respond to the agency. And then continue to engage with the community here in these foundations, also with your congressional leaders, in order to continue to drive awareness of the importance of this specific decision to the FDA.
Thanks, Dave. Okay. For the sake of time, I might skip a couple of these because I know they were already answered to some degree. If anyone within the community would like to have a specific answer to one of these that we skip over, please put it in the chat. We can answer all of the questions and send that out as a follow-up. Tons of questions coming in, which is awesome. But I will pull one from the Q&A box. So are you guys able to give us any information on how DCA affects ketone levels? We'll start with that one, and then I might pull one more out of the chat.
Sharon or Dave, do you want to take that one, or do?
Yeah, sure. So as Sharon mentioned, many of the patients but not all the patients were on a ketogenic diet in the study. I think it was probably on the order of 3/4, roughly, were adherent to a ketogenic diet throughout the study. And what we found is that in some patients, DCA led to an increase in ketone levels if they stayed on a ketogenic diet. In some cases, it didn't change it at all. And then what we saw is that, in certain patients, the diet was changed. So some patients may have had a 4-to-1 ratio on a ketogenic diet. And over the course of the study and the open label and the years that they were on medication, they may have liberalized that diet to include more carbohydrates. And so generally speaking, what I would say is there's a broad range of experience between DCA and that. There's no clear and obvious change that happens. That'd be one of those things if you are, regardless of if you're on a ketogenic diet or not, and DCA is something that you and your physician discuss as a treatment option, you would want to be monitored and monitor things like your beta hydroxybutyrate levels and ketone levels.
Okay, for the sake of time, I think we have about 7 minutes left. Francis, I'm going to have you go to the next slide, and we're gonna group some of these questions together. I think it's important as a PDCD community we rally together alongside SAIL and team. So a couple of the questions here: what can we do to help you get the medication approved by the FDA? And then what can parents do to help make this available for current and future patients? And then what happens next? So we have specific call to actions that we're going to end this meeting with to the community. But aside from the FDA type A meeting that we're pushing for, SAIL, is there anything that you would like to add specific from your side before we go into the next steps on our end?
I think the thing that I would just add is we mentioned we have an early access program. So there are a lot of people that seem to have questions that relate to, is there availability of medicine? How do I get on this? How do I do those types of things? So first of all, I'd say talk to your physician. And then if you think it's a good thing to attempt to do, it is available through the early access program if you have a genetic defect that's associated with PDCD. And it's not available everywhere. There are only a few institutions that do that, but there are programs that we're offering that can support travel and different things like that. So what I would say is if this is something that is important and is something that you're evaluating, talk to your physician, but know that that's there. And that would be something that SAIL is sponsoring currently.
And this is only in the US. Correct me if I'm wrong.
That is correct.
Okay, thank you for that. I know we had a couple of questions. Okay, Francis, I'm going to pass it over to you now since we only have 5 minutes left on specific call to actions for the group.
Thank you, Katie. Again, we apologize for the time constraints today, but on the last slide, I'm going to leave you with a contact from SAIL, Brian Nappy. So if you have additional questions, you can send them there, or if you have additional questions for the Hope for PDCD Foundation, I'm also going to put my contact there. So like I mentioned in the beginning, we as parents are not powerless. There are things that we can do to try to support SAIL and to support this treatment being approved at the FDA. So the first thing is you can share your DCA story on social media. Thank you to those of you who have already done so and continue to do so. We are going to follow up with an email to everybody that registered for this webinar with links to Canva templates or to the Ignite advocacy forum, et cetera, et cetera. You can look on the Hope for PDCD social media pages for examples to go off of. I recommend making a reel that posts or a post highlighting life with PDCD and what access to DCA means to you. And we have specific hashtags that we ask you to use. So hashtag approved DCA, hashtag Hope for PDCD, hashtag PDCD can't wait, and hashtag Hope with DCA. Please remember to tag us, the foundation, so that we can also share your content. And if you're not comfortable sharing on social media, that's totally understandable. And we are also going to provide a Google form where you can share your DCA story. And when I say DCA story, I mean that very broad in terms of whether you have participated in the clinical trial and you you have seen the treatment on your patient, or if you haven't and you're waiting for access. Secondly, we ask you to contact your elected representatives. Last I checked, the Ignite advocacy form, which thank you to UMDF and Mito Action for helping us circulate that, is up to over 2,000 signatures. You can also contact your local congressional office or senator's office and ask to speak to a health policy staff member, as they will be the most aware of the situation with the FDA and most aware of rare disease. When you call or email, remember to share your personal story and explain how PDCD impacts your family and why timely FDA action on DCA matters. Collectively, we are asking Congress to raise awareness about PDCD and DCA. And then remember to follow up with a thank you email, because it helps keep the conversation open. I just want to mention that we have a few board members that have had success at doing this. And if you need specific guidance, please email me afterwards and I can put you in touch with them and they can give you some specific advice. Reach out to local media. Hopefully everybody saw the wonderful TV interview that Lena O'Connor and Holly Solinsky did last weekend in Michigan. So reporters, like, this is a human interest story that reporters would be interested in hearing about, and they want to hear what life with PDCD looks like and what access to DCA would mean. And then finally, I'll end with stay in touch. So stay in touch with the foundation, because we're going to be continuing to post and raise awareness and put out these calls to action. Also, if you are in the clinical trial, stay in touch with your site coordinator and be responsive, because if the FDA requires further data collection, we want to get that to them in a timely manner. So I mentioned, if you have additional questions, which I know some of you do because we ran out of time, please just take a minute to jot down this contact information and we'll do our best to get to all of them. Thank you again to Saul and to everybody that participated today.
Thank you very much.